Hi. In this vodcast, we're going to be discussing photosynthesis. We'll talk about evolutionary adaptations of plants that have allowed photosynthesis to occur. We'll talk about what the implications of photosynthesis are, how structure and function relate, and the correlation between photosynthesis and cellular respiration. You'll see a lot of similarities here, so be sure to look for those parallels. Well, a great place to start is, of course, the beginning. So why photosynthesis? Bottom line, Earth is a solar-powered planet. What does that mean? It means that most life on the planet functions because sunlight comes in and provides the energy. In terms of energy, Earth has to be an open system. So we're constantly getting energy in because we know that as energy gets transformed, because it can either be created or destroyed, but as it's transformed and changes forms, the ultimate destination of all energy is heat energy, which isn't usable to life on this planet. Now there is some life that revolves around chemosynthesis instead, and we see this in places like the deep sea hydrothermal vents and other places like that. But for our purposes, most life on this planet revolves because of photosynthesis. So the producers, the autotrophs, have to do a few different things in order to fuel the planet. First and foremost, they have to collect solar energy and transform it into chemical energy, which is usable for other life. They have to also, number two, store the energy in a form that's stable, transportable, and can be moved from place to place. In order to do these things, they have to obtain the building blocks from the environment. Those building blocks are carbon, hydrogen, oxygen, nitrogen, phosphorus, potassium, sulfur, and magnesium. These are going to be incorporated into the macromolecules that can then be chemical energy for other plants or for other organisms. In addition, plants have to produce all of the molecules that they need for life. So not only are they engaging in photosynthesis, but we get a lot of the molecules we need by consuming food and by taking those parts and building them into new molecules. Well, plants need to do this too. They need to manufacture all of the proteins, lipids, and carbs. Well, plants need to do this too. They need to manufacture all the proteins, lipids, carbs, and nucleic acids that are necessary for them to continue on. Now, plant structure has evolved to support a number of different functions, and one of these is obtaining the materials from the environment that it needs to support photosynthesis. So let's look at some of the raw structures of this angiosperm in this example here. So we know that we need sunlight. Good for us, the plants come equipped with solar panels. Those leaves are really functioning as solar panels to collect sunlight and be harnessed by the chloroplast in order to engage in photosynthesis. We also need to get carbon dioxide in and get rid of oxygen gas. That happens through the leaves. Okay? And specifically in the leaves, portions called stomata. We'll talk about stomata a little bit more in just a moment, but the stomata are basically openings in the leaves that allow for gas exchange. We also need to uptake water, and that's coming in from the roots, and we've talked before about plant transport. And we need to get nutrients, which are also absorbed and hydrophilic and transported in the water and then into the plant via transport and osmosis diffusion, and then move through the plant as needed. So here we see the basic powerhouse of photosynthesis. Photosynthesis is occurring in the chloroplasts within these plant cells. Now the chloroplasts have a few distinct parts which are important for us to understand if we're to understand how the reactions that power photosynthesis actually occur. So the two main parts of the chloroplast is the stroma, which is right here in this picture, and is really kind of the space within the chloroplast. And then the thylakoids, here. And these stacks of thylakoids, when they're all piled together like a stack of pancakes, are called granum. These are the sites where the process of photosynthesis takes place. Now, chloroplasts, if you remember from our study of endosymbiont theory, are very similar to mitochondria. They have a double membrane, okay? One that's more closely eukaryotic in structure and composition, and one that's more similar to prokaryotic cells. They have their own DNA, and they're capable of replicating themselves. All of this points to the endosymbiont theory, which states that these were once free-living organisms that entered into a symbiosis with other cells. And this is very similar to what we believe happened with mitochondria. Now, the plant cells are packed full of chloroplast. And different kinds of cells will have different amounts of chloroplasts in different distributions. But chloroplasts are really the functional area, the functional space where photosynthesis is occurring. Let's go big picture for a minute and talk about what photosynthesis is in general. We know that in the process of photosynthesis, if we're to write out the chemical equation, our reactants are water and carbon dioxide, and our products are C6H12O6 or glucose and oxygen gas. Remember that we don't put energy in or sunlight in as a reactant because we know that matter can't be created or destroyed, it's also transformed, and what goes into a chemical reaction has to come out. But the energy is not a true reactant in this case. So, the first part of photosynthesis, called the light-dependent reactions or light reactions, takes place within the thylakoids. In this set of reactions, what we're going to do is we're going to take water in, so H2O is going in, and with the help of sunlight, that water gets split. When the water gets split, we release out oxygen gas. If we consider that for a moment, H2O yields that oxygen gas. The question is, what happened to the hydrogens? The hydrogens go into a molecule called NADPH. That should look a little bit familiar. If you remember in photosynthesis, we talked about NADH. And if you recall, that was a high energy carrier. Think of it as a set of hot mitts, transferring something hot from the oven to the table. They're transporting the energy for us in this case. It's money in the bank. Also out of these light reactions, we produce a small amount of ATP. Now both ATP and NADPH, which are produced in these light-dependent reactions, are then going to travel to what we call either the light-independent reactions or the Kelvin cycle. And that occurs in the stroma of the chloroplast. Now, remember, the point of photosynthesis is to make sugars. Have we done that yet? The answer is no. There's also another reactant we haven't used yet. We have not utilized carbon dioxide. In the Kelvin cycle, the carbon dioxide enters and through a series of different chemical reactions is catalyzed, and with the help of the NADPH, the energy from that, along with that hydrogen, which is now being shuttled as well, we're going to produce sugars, C6H12O6. As a byproduct of this process, we're going to break apart the NADPH and the ATP. But fortunately for cells, we're really good at recycling materials, so those aren't completely lost. Rather, we end up with NADP plus and ADP and a phosphate. So we've taken the energy from the ATP, and from the NADPH, we've shuttled the electrons there, along with a hydrogen, to combine and make the molecule glucose. Let's take a look at the balanced equation here. We're inputting 6 CO2 and 12 H2O, getting out glucose, oxygen, and some amount of water. Now you may have seen this reaction room without water being a product and only having 6 H2O on this side. Okay? And that's a simplified form of this chemical equation. But the reality is water goes in and water is produced as a byproduct. In reality, we're not really producing glucose in this. Okay? We're producing a molecule called G3P, and that should also look really familiar. Here's another one of those correlations. That was a molecule that glucose was broken down into in the process of glycolysis. G3P is a 3-carbon sugar that's used to synthesize glucose and other fuels and other carbs. Let's review for a second. Where did the carbon in this glucose molecule come from? That's right, if we look down at our equation here, that C comes from the molecule CO2. Now how about the oxygen in O2? This is a tricky one. Make sure that you remember that that comes from the splitting of water in those light-dependent reactions. And we know this because when plants were given radioactive water with radioactively labeled oxygen, and other plants were given carbon dioxide with radioactively labeled oxygen, the plants that were given with the radioactively treated water, the oxygen gas that was produced was also radioactive. So that is a little insight into how we've actually scientifically proven where these molecules come from. Here's another really interesting piece about photosynthesis. The chloroplast that are engaging in photosynthesis, that are really the machinery of this process, okay, are green. We know that things are the color they are because of the wavelengths of light they reflect. Chloroplasts are absorbing light energy and using that to convert it into a chemical energy. So the question is, what colors of light, what energies of light are chloroplasts really efficient at grabbing onto? And the answer is in the pigments that are found in the chloroplasts themselves. So take a look at this graph here. Really elegant experiment. If we look at our first figure here, we can see the absorption of light by the chloroplast, different pigments. In the chloroplast, we have chlorophyll A, chlorophyll B, and these carotenoids. And each one of these different pigments has its own range of wavelengths of light that it's optimized to absorb. Okay? We can see that chlorophyll A peaks right about, if we follow down here, right about 430. We can see that chlorophyll B has a slightly longer wavelength of light that it absorbs. And we can also see that there's a whole other area up here that they absorb. So we kind of have this bimodal distribution of different pigments, of different wavelengths that these pigments are able to absorb. In this really elegant experiment, we can see that the optimal wavelengths of absorption are also correlated to the optimal amounts of rate of photosynthesis, and photosynthesis being measured by the amount of O2 produced, which is a byproduct. And in this particular experiment, in Engelmann's experiment, what he did is he took this filament of algae, this spirogyra, okay, and exposed different sections to different wavelengths of light. And there are aerobic bacteria, meaning that they require oxygen in this aquatic environment. And the aerobic bacteria multiplied and increased and were present in higher population numbers in the areas where there was increased photosynthesis, because in areas of increased photosynthesis, we'd have increased oxygen production. And if oxygen is a necessary component for those bacteria, then it follows that they would exist where this was present in abundance, and they would exist in proportion to the amount of oxygen present. Let's go back full circle here for a moment. A key piece to how cellular respiration occurred was really in the electron transport chain found within the mitochondria. Remember that glycolysis was breaking things down and produced a small amount of energy. But what was really the biggest bang for our buck as we're going through this is we're producing high energy carriers, NADH and FADH2. Those high energy carriers are shuttling electrons and hydrogen ions to the electron transport chain. As those electrons moved through the electron transport chain, so if we remember down here that NADH was this high energy carrier, and it's donating its electrons, and as those electrons move down from one protein to the next, each one a little bit more electronegative, each one a little bit more electronegative to pull that electron along, that movement of the electrons was being used to do work. And the work it was doing was this transport work of pumping hydrogen ions across the membrane. That built up a hydrogen ion concentration that, when the hydrogen ions were allowed to flow back down through ATP synthase, that energy could be harnessed to take ADP and a phosphate and attach them to make ATP. So it was the NADH and the FADH2 that were really driving this whole process. Remember, at the end of the electron transport chain, oxygen was there to accept those electrons. It was the final electron acceptor because oxygen's the biggest bully on the block. It's one of the most biological electronegative elements, and so it's absorbing that, or taking on that last electron. It's being neutralized essentially by the addition of hydrogen ions, and hence we end up with water. Now, something similar is happening in photosynthesis, so this should look really familiar to us. Where the electrons are coming from and what is powering the system are the differences here. But essentially, big picture here, in photosynthesis, we have light energy coming into the system. Okay? That light energy is allowing water to be split. When the water is split or broken apart, we end up with oxygen, hydrogen ions, and what's missing? You guessed it, electrons. Okay. The electrons are going into what we call photosystem 2 here, and we'll explain more of the details in a minute, but let's just stay big picture. And electrons are essentially being passed down this transport chain. As they're passed down the transport chain, they're doing some work. Some of the work is in moving hydrogen ions into this thylakoid space and setting up a concentration gradient. That concentration gradient is going to be used right here. Again, this should look really familiar. Where as the hydrogen ions flow down through ATP synthase, that energy will be harnessed to take ADP, bond it to another phosphate to make ATP. This is going to then go on to the Kelvin cycle in just a moment. As those electrons are moving down, eventually they enter a second photosystem, which is photosystem 1. Okay? Photosystem 2 is the first in the series, photosystem 1 is the second, because they were named in the order that they were discovered. We're recharging and using light energy again here. And those electron movements down the second piece of the electron transport chain here are going to end up with a very special enzyme. That's NAD plus reductase. NAD plus reductase. Remember, in a reduction, if a molecule is being reduced, it's gaining electrons. So NADP plus is going to take NADP plus and add the electrons from this electron transport chain to it, along with hydrogen ions, so that we have a net neutral molecule at the end to produce NADPH. NADPH is then going to go on along with ATP to the Kelvin cycle. One way to keep this high energy carrier straight is that NADPH has a P in it for photosynthesis, whereas NADH is just in cellular respiration. Let's look at some of the similarities here again across the reactions. We're really looking at photosynthesis versus cellular respiration. Photosynthesis is going to be powered by sunlight, where respiration is going to be powered by the breakdown of that glucose molecule. But in both reactions, we move electrons. We move electrons through an electron transport chain. In both reactions, the movement of electrons runs a hydrogen ion pump that pumps the hydrogen ions, or we know that H plus is essentially a proton. This pumping is going to build our concentration gradient across the membrane. When we build the concentration gradient across the membrane, the hydrogen ions naturally want to flow down their concentration gradient, which is this driving flow of protons, and that flows down through the only available route, which is ATP synthase. As they flow down through ATP synthase, the energy is harnessed and bonds a phosphate group to ADP to make ATP. Lots of similarities here. If you keep making these connections between the two reactions, it's going to make both of them simpler to understand and help you to really understand the connections and applications going forward. Let's now take a closer look at how we are actually fueling this process and fueling the electron transport chain. Photosystems. We have two different photosystems that are part of the electron transport chain. And their function is really to gather that solar energy, gather that light energy, and excite these pigment molecules of chlorophyll that are present in the photosystems. This is going to allow us to excite electrons and send electrons through the electron transport chain. So this photosystem as a whole, if you look closely at it, consists of a reaction center complex, which is right here, and these light harvesting complexes that are full of these pigment molecules. These pigment molecules are really going to take the energy from the sunlight and transfer it to this reaction center. Primary electron acceptor that's found here in the reaction center complex is going to accept the accepted electrons from this special pair of chlorophyll A molecules here. So essentially, the sunlight is going to come in. That sunlight excites the chlorophyll molecules. When we excite molecules, what we're really doing is talking about exciting the electrons. As the electrons get excited, they jump up to a higher energy level, and we're going to be almost like a series of dominoes here, where the excited electrons are exciting the next chlorophyll molecule and the electrons in it, and exciting the next chlorophyll molecule and the electrons in it, and so on and so forth, until we get to this special pair of chlorophyll A molecules. And once we get there, the electrons get so excited, but they don't come back down. In each of these first molecules, the electrons get excited, they come back down as they lose their energy, and that energy gets passed on to the next chlorophyll molecule. But this special pair of chlorophyll A molecules here, the electrons get excited, and then they lose their electrons. The electrons get passed on to this reaction center complex, this primary electron acceptor. That does create a little problem, because now we've got a hole here. Now they're missing electrons, and we're going to have to find a way to fill those back up. So, photosystem 2 is the first one in this system, but it's named photosystem 2 because it was the second photosystem that was discovered. And in photosystem 2, we have the pigment chlorophyll A. And chlorophyll A, this P680, means that its peak absorption level is at a wavelength of 680 nanometers. That's a red light wavelength. And it's going to generate energy in the form of ATP. So the electrons here and the energy here is going to be used to pass the electrons down the electron transport chain, to drive the hydrogen ions across the membrane, to build up the gradient, to then allow them to flow back down through ATP synthase to produce ATP. That's the purpose of photosystem 2. It's passing those electrons along to create that gradient. We'll talk about where those electrons end up in a minute. The purpose of photosystem 1 is a little bit different. Photosystem 1 is the second one in the system, but it was discovered first. It has chlorophyll B molecules, and its peak absorption is a little bit different. It's at 700 nanometers, which is still in that red wavelength. The purpose of photosystem 2 is to pass the electrons along until it gets to the NADPH reductase, which is going to use those electrons to take NADP+, as well as hydrogen ions and electrons, combine them together to make NADPH, which is going to go on to fuel the Kelvin cycle. The electrons from photosystem 2 get passed on to photosystem 1. Let's take a look at what this looks like all together. So, we have light energy coming in here. That light energy coming in is going to excite our chlorophyll molecules. As those chlorophyll molecules get excited and their electrons fall back down into a stable state, that energy is getting passed on to the subsequent chlorophyll molecules. This is what's happening in both of the photosystems. That continues to happen until that energy reaches this special pair of chlorophyll A molecules, P680, here in the reaction center. As those molecules get excited and their electrons get excited, they get passed on to that primary electron acceptor, and you can see that that electron has been passed on here. So now that there's a hole there, they've lost their electrons. They've passed them on to that primary acceptor. This is where water comes in. Water is going to be split in photosystem 2. As the water is split, we're going to strip off 2 electrons. Okay. Remember that a hydrogen ion, or a hydrogen atom, is essentially proton and an electron. So if I've produced 2 electrons and stripped 2 electrons away, I also have 2 hydrogen atoms, this hydrogen ions that I've produced. There they are. What's left of H2O? The oxygen gas. These electrons that are stripped off of water are going to go down and replace the electrons that special pair of chlorophyll A molecules lost, that P680. So in this way, there's now 2 more electrons ready so that as the system continues, the electrons can get excited again. Otherwise, you're stripping away all the electrons out of this chlorophyll molecule and the system won't be sustainable. So we have to constantly replace those electrons as the system progresses. Now, those electrons that have been excited to that primary electron acceptor, okay, are going to get passed down the electron transport chain. Cytochrome C is a really important part of this, and it's a molecule that's highly conserved across all life. And in fact, we can figure out the evolutionary clocks and how fast things have evolved by sequencing cytochrome C and seeing the similarities and differences between the same molecule in different organisms. We're driving hydrogen ions across the gradient. That's going to allow us to then manufacture ATP as those hydrogen ions flow back down through ATP synthase. These electrons are going to continue on until we get to photosystem 1. In photosystem 1, we have sunlight coming in again, and as that sunlight comes in, it excites the electrons in the chlorophyll B molecules this time. The chlorophyll B molecules electrons get excited. As they fall back down, the energy gets passed on to the next and the next and the next until we reach that special pair of chlorophyll B molecules, this P700, here in the reaction center complex. Their electrons get excited. They go up to that primary acceptor. They don't come back down. Water replaced the electrons here for P680. What's replacing the electrons here for P700? That's right. It's the initial electrons from that electron transport chain. The electrons that are excited out of P700 get excited, go to that primary acceptor. Now they flow down the electron transport chain here until we reach NADP plus reductase. NAD plus reductase is going to take those electrons, bind them to hydrogen ion, and NADP plus to make the high energy carrier NADPH, which is then off to the Kelvin cycle. Here it is one more time. The process as a whole. Light energy comes in, excites the electrons, makes it to the primary reaction complex right there in the center. The P600 chlorophyll A molecules get excited, send their electrons onto that primary electron acceptor. They get passed down the electron transport chain and are doing work, pumping hydrogen ions into that thylakoid space, building up the gradient to go through ATP synthase to manufacture ATP. At the same time, in photosystem 1, light energy is coming in, exciting the electrons. The special pair of chlorophyll B molecules is getting excited, passing its electrons on to the primary electron acceptor in photosystem 1. P700's electrons are replaced by the electrons from photosystem 2. The electrons that are excited go through until they reach NADP plus reductase, which uses those electrons and binds them to NADP plus and hydrogen ion in order to make NADPH, which is a high energy carrier, and it will fuel the Kelvin cycle. So let's do a quick summary here. Where does the energy come from in photosynthesis? That's right. Energy in photosynthesis is coming from the sun, or light energy. Where do the electrons come from? The electrons originally are going to come from the splitting of water. Remember that splitting of water in photosystem 2 is going to replace the electrons that are lost in that first photosystem. Where's the water come from? Well, you're getting the water from the roots in the plant. And where does the oxygen come from? Remember that oxygen comes from the splitting of water. And we know this because we've radioactively labeled it and seen its progression. Where's the O2 going to go? Remember those specialized structures in the leaves? That's right, the O2 is going to pass back out of the leaves through the stomata. Where does that hydrogen ion come from? Again, the hydrogen ion comes from the splitting of water and is going to be used to build up that concentration gradient to drive the production of ATP. I guess we've answered that one. Where does the ATP come from? The hydrogen ion gradient that's established due to the electron transport chain fueled by photosystem 2, which gets its energy from the sun, and the electrons from water. And that ATP is going onto the Calvin cycle, and it can also be used to do work. The NADPH is a direct result of photosystem 1, which uses the electrons to drive NADP plus reductase to manufacture NADPH from those electrons, NADP plus, and hydrogen ions. And that NADPH is going to be used to fuel the Calvin cycle. That's the big overview. The next part is going to be to take a closer look at the Calvin cycle. The Calvin cycle is the second part of photosynthesis, and it really has 3 phases. What's important for you to remember is what's the general purpose of each phase, what goes in and what comes out. In phase 1 of the Calvin cycle, and remember that the whole Calvin cycle takes place in the stroma of the chloroplast, the first phase is called carbon fixation. And carbon fixation is when we really take that CO2 and incorporate it into an organic molecule. And this is catalyzed by the enzyme Rubisco, which is probably one of the most abundant proteins on the entire planet. The second phase of the Calvin cycle is reduction. In reduction, we're actually going to produce the sugar. It's that G3P sugar, which is the intermediate before we actually can manufacture glucose and other sugars. For every 3 CO2 molecules that go into the Calvin cycle, 6 G3P are produced, but 5 of those get recycled and go on to phase 3. So our net gain is really 1, which makes sense if we count the carbons. 3 carbons are going in, so I should only get 1 molecule of a 3-carbon sugar coming out. Those 5 other G3P molecules go on to regeneration. In regeneration, those molecules are rearranged, their structure is modified, so that we can go back and start the process again. In this, RUBP is going to be regenerated from these 5 G3P molecules. So let's take a look at this Calvin cycle one piece at a time. Now, initially we have our 3 carbon dioxide molecules entering here. Remember that the first portion of the cycle, phase 1, is carbon fixation, where the enzyme Rubisco is going to be used to take those carbon dioxide molecules and now fix the carbon into an organic molecule, which will eventually end up as our G3P sugar. This happens through a series of reactions, each catalyzed by a different enzyme, where we're adding phosphate groups by using ATP and the NADPH. We're also adding in the hydrogen ions out of that NADPH to end up manufacturing G3P, okay, that G3P sugar. And that comes at the end in phase 2, that reduction. Now, here it shows us that we produce 6 G3P molecules. Well, one of those goes on and exits the Calvin cycle as actually G3P, which can then be used to manufacture glucose and other sugars. That leaves us with 5 remaining molecules. Those 5 remaining molecules are now recycled through regeneration. What are we regenerating? We're regenerating RUBP, which is that acceptor of the carbon dioxide molecules. So, we regenerate again through a series of enzyme-catalyzed reactions. This requires energy because we're making some new bonds and rearranging these, and we end up with RUBP. RUBP is used by Rubisco to fix that carbon back into phase 1 of carbon fixation. So each turn of the Calvin cycle produces 1 G3P molecule and uses 3 carbon dioxides. If we go back to that photosynthesis equation, We know then, in order to produce one molecule of C6H12O6, how many times does the Kelvin cycle need to turn? That's right, the answer is 2 times. And it will consume a total of 6 carbon dioxide molecules, 3 in each turn, to produce 2 molecules of G3P, which will then produce 1 molecule of C6H12O6. That's it for kind of the overview of photosynthesis, what's happening where and why. Now there are some variations on this theme in terms of different plants and their evolutionary adaptations, and we'll discuss that at another time. Thanks!